getting the real, like, digital display of the pH using your pH meters. So one of the things then, let's just go straight to what you're going to do. So your indicator actually is going to be a molecule in red cabbage that makes indicators are great. We'll be talking more about indicators that indicate pH and acidity, basicity in lecture coming up. But the thing about it is, if we look closer, let's see if it'll do this for me. Now I need to go this way. Awesome. So that is step numbers 1 and 2, sort of. The rest of step number 2 says this. You are going to be using, when it says large test tubes, I just want you to go ahead and help yourself and grab some test tubes that are drying over here. You don't need the big, humongous ones. This is the 150 millimeter test tube, so don't grab the humongous big ones, okay? Step number 2 says you're going to put 2 mils of your cabbage extract. Now, over there by that rightmost balance, I put the pipettes that we're used to using. Remember that the smaller or narrower pipette actually has gradations on it that is 1 mil. So I would use 2 squirts of that actually from your red cabbage juice into these. You're going to use, so you'll have 2 mils in 5 test tubes. Test tube racks are in the middle hood. Help yourself, like I said, to these nice clean test tubes that I clean. Then it says you're going to use these buffered solutions. So I have buffered solutions here of 3, 5, 7, 9, and 11 under the hood. We're going to talk more about buffered solutions, and some of you already know how this works. But buffered solutions, the word buffered means that they fight against pH change. So that's the cool thing about these. We'll be kind of talking about what salts go in those to make those buffered at those particular pHs coming up probably next week. So the point is that you'll need to identify one of your 5 test tubes as pH 3. I did go ahead and I never did get around to ordering some more adhesive labels. Adhesive labels are my favorite. So actually I snagged some of these from the stationery stuff. I think they'll work pretty good. So mark your test tubes pH 3, 5, 7, 9, and 11. Then you're going to go ahead and take 8 mils of the buffer solution and add it to your 2 mils. Now what I would do is go ahead and I would use a graduated cylinder. I know you're going to have to rinse out probably 3 times or so between your pH, your different buffered solutions, but that's what I would use to get your 8 mils. Add 8 mils to your 2 mils, put it in your test tube rack, and that is your standard. Okay, it's your visual standard. So that's step number 2. And then step number 3 is you're ready to basically, no pun intended, do a similar thing to those 7 clear solutions that are in the middle hood. Okay, what is that? So basically you'll need another 7 test tubes. Help yourself to the test tubes. We got plenty if we run out. To each of those 7 test tubes, put 2 mils of indicator in each of those 7 test tubes. Okay, and label them 1 through 7. And I want to push this out to Twitter to estimate the pH of your, I guess it's just there in step number 3. Okay. Compare the color and the intensity of the solutions. By the way, you should be writing, you'll write this down in your notebook. Write down the color of what you observe and see which of your standards it looks most like. Okay, and that's your estimated pH. Now when you're done with that, before you dump anything, I want to see what you have. Okay, before you dump anything, I want to see what you have. I want to grab a picture and I want to just make sure we're all on the same page. Okay. So once that's done, what you're going to do is you're going to dump out all of your test tubes. I say this because sometimes students like to go on to the next part with their test tubes. You will not use your test tubes for the second part. Okay, for the second part, it's kind of not much to be said about it. All the second part is in number 5 right there. So for the second part, you're going to turn your attention to your lab, your whatever these are. Yeah, lab quests. I guess they are lab quests. And I kind of played around with it. This is the first time for me to use them. So if you kind of look here, we've not done pH measurements. Maybe we did pH measurements in Chem 1, I thought. Maybe we didn't. So here's the deal. You're going to be using, the probes are on those long boxes right there, and you can see that I'm actually going to use, you're going to want a ring stand and go ahead and use a ring clamp or a clamp on your ring stand. You guys are going to be using these for next week's lab too. Now the cool thing about this pH probe, right, is if you can see that little glass thing in there. I know it looks like solid glass, but actually it's a semi-permeable membrane, a selective membrane. It's selective just for the hydrogen ion. Is that not cool or what? You can actually get different electrodes where the membrane is selective for different ions. This one's the hydrogen ion. So it used to be, and these are awesome. Dr. Snipes and I both think these are awesome. It used to be that actually when you put this in your liquid to go ahead and get kind of the hydrogen ion to equilibrate past that membrane, you had to allow a little bit of time. You still do, but these are pretty darn fast. Okay. The other thing is when you get this out of the box, you're going to see that this is stored in liquid. You're going to see it's in one of these things. The thing is that really selective, ion-selective membrane, it's very sensitive to being dehydrated. It ruins it. So this is actually just a storage solution. So these are in your boxes. To get them off, I know you guys will figure this out, but basically you unscrew this to loosen it, and it will just slide out. Well, after you're done, we're going to put it back in that solution. The other thing is, did you notice while I wasn't using it, I had it sitting in a beaker over there? That's just tap water. So I didn't want it to dehydrate between when I played with it earlier and when you guys were going to see it. What I would suggest, that's a good kind of habit to get into, if you're going to do some readings, because you guys are going to do 7 readings, basically you're going to read those 7 solutions. What I would do is have kind of a, get some DI water, that's what this is, okay, something to kind of store in between, okay. The other thing is you might want to have a waste beaker. The 400 ml beaker, which you're not going to use for your red cabbage, works great for kind of a waste beaker. You're going to need to kind of douse off the thing in between time, the thing being the electrode. So that's kind of why this ring stand is kind of handy. If you're the only one there, you just kind of go like this. Your instructions might say that you're supposed to dry this between samples. It says use something like Kim wipes. I don't have any Kim wipes right now. But if you're just careful with this sort of towel, you should be fine. One of the things is you don't want to kind of induce some sort of electrical charge on it by rubbing it, right? So you just want to kind of pat it between your samples, and then you're ready to go on the next sample. Let me double check what these new instructions say. These new instructions should say basically you're going to test your probe, and remember these are so darn smart. When you plug this into here, it knows it's a pH meter. That did not used to be the case. Okay, your instructions say something like basically grab one of these buffered solutions. I'm going to grab 5. Actually, I'm going to grab another one. I have dedicated beakers up here, and I think we're such a small group we don't have to. You guys should be able to use that. I'm going to grab 9 and check your meter, and if it's on, you don't have to calibrate it. If it's off or not on to your satisfaction, you need to go through the calibration procedure, and it's really not too bad. It's indicated in here. So I'm just going to come over here and wake up my LabQuest. It's already got my probe there. I've got this rice and ready to go. Remember, you have to click the app and douse it in there. Next week, when we use your pH probes, we're actually going to put a stir bar there to kind of circulate things, because you guys are going to be doing titrations. But a lot of times, if you're measuring pHs, you kind of want to get the liquid past the membrane, so a lot of times you'll kind of go like that. For some reason, I used to do this a lot in my environmental lab days. Sometimes solutions take a long time to stabilize. This one's saying 9.04, and this is buffered solution 9. I can live with that. Okay, so for me, I would be ready to measure my 7 samples. Okay, so I'm going to go ahead while I'm thinking of it, set this aside so somebody else can use this. It's labeled 9. I'm going to douse this down. I'm going to put it in kind of a storage solution. So again, you're not using your test tubes, you're not using red cabbage, you're not using anything like that for your second part. So for your second part, what I have in mind this semester is for you just to basically go ahead and measure it out of here. So you guys, if you all get here at the same time, that's no problem because there's 7 solutions, okay? You need to pour the Sprite up here, you need to pour this cleaner into here. I think you guys kind of got the idea. It's a fun lab, we do need to wear goggles. And so let's look at the calculations, okay? So the calculations say make a table of your 7 solutions. and rank them from the most acidic to the least acidic. Okay, that's the lowest pH to the highest pH. And of course, make sure you say which solution is which, and make a column to say, is it acidic, is it neutral, or is it basic? And then I would, on that same table, well, maybe not, because no, you're going to have to show me how you do it. It can't be on the table. You can add it to the table later. For calculation 2, you need to show me how you take the pH, and that would be the pH that this said, not your estimated pH. pH from part 2. You're supposed to take that pH value for that same solution, also calculate the POH, which is like a difference from 14, right? And the hydrogen ion, the molar concentration of the hydrogen ion, the molar concentration of the hydroxide ion. You must show your how you do that for each one of them. And you can throw them back up in the table that you prepared in 1 if you want to. Be nice and clean and neat. Okay, so that's all I got. I'll meet you on the other side. Here are the 7 solutions that you're going to be using. Okay, the first one is tap water, the second one is vinegar, household ammonia, shower cleaner, Sprite, Alka-Seltzer as required to be prepared, and water from the drinking fountain. Okay, so those are the 7. Okay, so the deal is you have 7 test tubes and 2 mils of indicator, and then you put 8 mils of each of those in there. So you're going to have a test tube rack with 5 and 7, right? Okay. So then you're ready to, like it probably, use your standard buffered solution.